Okay, great. Then we shall continue our yesterday's talk on probiotics, prebiotics, and postbiotics. And do you have any a question before I start the talk?
No, sir.
Okay, then we shall go to the slide. Yesterday, I showed this slide. Can you see the slide?
Yes, sir.
Yes, sir.
Yesterday, we discussed that plants also have a digestive system. Soil, uh, in the soil. The microbes in the soil are as vital or more so for plant nutritional needs as for human digestion. Biologicals are yogurt for plants, trees, and turf. That means they are also called biologicals. This is another very interesting term. Biologicals. That means living organisms. If you use them as a technology, then you can call it biologicals. Uh, Probiotic technology also newly people call them biologicals. Prebiotics. Yesterday we discussed a lot about probiotics. Today we shall discuss on prebiotics. In 1995, prebiotics were defined by Gibson and Robert Freud as non-digested food components that, through the stimulation of growth and or activity of single type or a limited amount of microorganisms residing in the gastrointestinal tract, improve the health condition of a host. That means prebiotics, they are non-digested food components. Our digestive system cannot digest them for our own nutrition, but they are useful for the microorganisms, that is, those who are helping us for digestion as well as for immunity, that is probiotics. So the food of probiotics, we can call them prebiotics. In 2004, the definition was updated and prebiotics were defined as selectively fermented components allowing specific changes in the composition and or activity of microorganisms in the gastrointestinal tract, beneficial for host health and well-being. Very interesting. That means fermented components, selectively fermented components, allowing specific changes in the composition and activity of microorganisms. That means that those components you use to increase the activity of or change the activity of microorganisms that are residing in the gastrointestinal tract. Uh, Ultimate target is to use the prebiotics to offer the benefit to the host health or well-being. It can be, not only human. It can be in case of animals, in case of plants, or any other eukaryotic organism. Finally, in 2007, FAO experts described prebiotics as a non-viable food components. Prebiotics are non-viable food components that confers a health benefit on the host associated with modulation of the microbiota. That means a very simple definition. Prebiotics has some special criteria. They are non-viable food component and they are able to confer health benefits to the host by they can modulate, that means change the microbiota, maybe the population and their activity. There is another term called Synbiotics. In 1995, Gibson and Robert Freud introduced the term symbiotic to describe a combination of synergistically acting probiotics and prebiotics. That means symbiotics are synergistic, synergistically acting probiotics and prebiotics. A selected component introduced to the gastrointestinal tract should selectively stimulate growth and or activate the metabolism of a physiological intestinal microbiota, thus conferring beneficial effect to the host health. As the word symbiotic implies synergy, the term should be reserved for those products in which a prebiotic component selectively favors a probiotic microorganisms. The principal purpose of that type of combination is the improvement of survival of probiotic microorganisms in the gastrointestinal tract. Synbiotics have both probiotic and prebiotic properties and are created in order to overcome some possible difficulties in the survival of probiotics in the gastrointestinal tract. Very interesting. You can see Synbiotics have both probiotic and prebiotic properties, and they are actually involved in uh, enhancing the survival capacity of the probiotics in the gastrointestinal tract. Here, there are some selection criteria of probiotic strains, how we can select a probiotic strain. This is very, very important. The most important criteria is safety. The second criteria, probiotic must be functional. Third criteria, technological usability. So it could be used as a technology. So these three criteria are very important for selection of the probiotic strain. First is safety, human or animal origin, required properties. There must be human or animal origin in case of human or animal probiotics. But you can add here human or animal or plant origin because in case of plant, the probiotic, this term we are now using frequently. Isolated from the gastrointestinal tract of healthy individuals. Healthy individuals, gastrointestinal tract of healthy individuals, and you can add another point in case of plant, isolated from the internal tissues of the healthy plant. Okay, history of safe use. It should be safe use. If has the history of, because such species or strain has history of safe use, then it is. That means its safety must be ensured. Precise diagnostic identification, phenotype and genotype. That means it should be identified very precisely, not only phenotype but also genotype. In case of bacteria, we use 16S rRNA gene sequencing, or we can use whole genome sequencing to precisely identify the probiotic organism. Absence of data regarding and associated with infective disease. There should not be any report that it is causing the disease to other organisms. Then you can use it industrially as a probiotic. Absence of the ability to cleave bile acid salts. No adverse effects. Absence of genes responsible for antibiotic resistance localized in non-stable elements. This is very, very important. So you have to check whether the genome of this candidate probiotic has no genes responsible for antibiotic resistance. That means at this point, it is clear that we have to do some molecular studies to confirm that it has no harmful effects to the environment. It has no harmful effects to the non-targeted organisms. So this type of safety we have to maintain before we consider a strain as a probiotic for any for human, animal, or any other useful eukaryotic organisms like plants. Second is functionality. Functionality, competitiveness with respect to the microbiota inhibiting the intestinal ecosystem. This is most important because in the intestine or in the rhizosphere or in the phyllosphere of plant, there are lots of micro biota. There are established microbiome structure. When you incorporate your candidate probiotic strain, it must be competitive. That means it can colonize their uh, fighting with the inhabiting organisms to exert the beneficial effect. Ability to survive and maintain the metabolic activity and to grow in the target site. For example, if you apply in the intestine, gastrointestine, it has to ability to survive there and maintain a metabolic activity, normal metabolic activity, and grow in the target site. That means the site you applied. For example, in case of plant, if you want to apply it in the leaf, then it should colonize and survive and grow well with normal metabolic activity on the leaf surface or even in the root or even internal tissues. Resistance to bile salts and enzymes in case of human probiotics or animal probiotics. Resistance to low pH in the stomach. Competitiveness with respect in case of plant, if you want to apply it in the saline area or acidic soil or drought, it must resist those important abiotic stresses. Competitiveness with respect to microbial species inhabiting the intestinal ecosystem, including closely related species. Antagonistic activity towards pathogen. This is very, very important. Antagonistic activity towards pathogen. For example, Helicobacter pylori, Salmonella, Listeria, Clostridium. These are very notorious pathogens recognized in the intestinal or guts, and you apply probiotic should have ability to antagonize them. Antagonize means they can produce antibiotics, or they can produce enzymes to kill them or to suppress their proliferation or somehow antagonize them. Resistance to bacteriocin and acids produced by the endogenic intestinal microbiota. Adherence and ability to colonize some particular sites within the host organisms and appropriate survival rate in the gastrointestinal system. So these are the functionality. For example, in case of plant probiotic, they might have the ability to promote the growth of plant. Shafi, you can add the additional points. In case of plant probiotics, they might have the ability to fix atmospheric nitrogen. They might have ability to produce phytohormones and other substances to promote the growth of the plants, or they might have the ability to dissolve the insoluble uh, uh, nutrient elements in inorganic and organic forms, and they might have in both cases might have ability to induce systemic resistance in the host plant. That means boost up immunity of the host. So these are the functionality you must ensure. Not necessarily all those uh, are uh, 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 are present in the specific uh, uh, probiotic strain, but these are the points you must consider before selecting a suitable probiotic for technological development. Technological usability, easy production of high biomass amounts and high productivity of cultures. If it is unculturable and it is no meaningful, so you have to find the suitable media for its culture and huge large scale production. Viability and stability of the desired properties of probiotic bacteria during the fixing process, that is freezing, freeze drying, preparation, distribution of the products. That means if you make a capsule or if you make a tablet or if you make in powder form, its shelf life should be enough. High storage survival rate in finished products in aerobic or micro aerophilic conditions, and a guarantee of desired sensory properties of finished products in the case of the food industry. Genetic stability. Genetically, it should be stable, because otherwise, in case of many probiotic candidates, we found that if we store in the freezer, after 3 months, when we want to recover, we see it genetically changed and no activity is remained, and it is a big problem. So it should be genetically stable and resistance to bacteriophages, because virus are the dangerous enemy of the uh, probiotic organisms. So bacteriophages should be resisted by the your target probiotic bacteria. And in that case, maybe CRISPR-Cas system, yeah, if it is functional in your bacteria, then it would be smart enough to tackle the bacteriophages. So this is the selection criteria of probiotic strain, and discussed, and this is very important point to be considered for selecting a probiotic and discovering and commercializing a probiotic candidate. Probiotic microorganisms used in human nutrition, let us see some of them. For example, most of them you see lactobacillus, lactobacillus acidophilus, lactobacillus amyloborus. So there are many lactobacillus. And type of bifidobacterium, there are some bifidobacterium also, bifidobacterium adolescentis, animalis. So bifidobacterium and lactobacillus, they are very famous in case of human as well as animal nutrition. Other lactic acid bacteria also used in human nutrition: Enterococcus, Faecium, Lactococcus, Lactis, and Streptococcus thermophilus. And other microorganisms, except Lactobacillus, Bifidobacterium, and other bacteria, other microorganisms like Bacillus, Clausi, Escherichia coli, Nisel 19 uh, 17, it is another strain, and Saccharomyces cerevisiae, this is not the bacterium, it is a fungus, because Saccharomyces cerevisiae we use to produce the yeast. It is yeast, common name is yeast, and yeast has been used as a biotechnological product in baking and other purposes for a long time. So these are some of the examples of probiotic microorganisms used in human nutrition. There are some more If you search the literature, you can find it. So this is all for today, and we shall discuss more. You can ask some question.
Sir, I want to ask one question.
Sir, you have mentioned that the
One by one. First, Sorwardi is asking the question, that is Shafi, and then Ruhul Amin, you will ask the question.
uh, so uh, prebiotic and probiotics, the combination is also important. So it is also advisable to all because we need a doctor's suggestion or something like that to take this additive.
You mean prebiotic and probiotic competition. Compatibility is very important because we use prebiotic. There are non-living substances like polysaccharide or like that to promote the growth of the probiotic. So in that case, they should be tested and there should be, wide uh, uh, choice of the microorganisms. Otherwise, the prebiotics would not serve the purpose, and they must be safe for the probiotic microorganism and loved by those organisms to use them as their nutrition, as their growth promotion and others. You are right. When we make industrial formulation, in that case prebiotic also used in the formulation, so that initially a probiotic organism, when they landed in the target site, they can get enough good environment, that means nutrition and other supports from the prebiotics for their proliferation and to exert their target function. Shafi, any more question?
No, sir.
Okay. Next, Ruhul Amin.
uh, yes, sir. You have mentioned that bacteriophages. These are actually, I know that these are viruses, and they destroy the beneficial bacteria. In case of doi, we know that doi sometimes becomes sour taste, and bacteriophages uh, engulf the beneficial bacteria from this. So you mentioned that we can use CRISPR generated. So how can we use CRISPR in the target bacteria to protect from um, bacteriophages?
A good question. Actually, when CRISPR was discovered, CRISPR-Cas system was discovered as adaptive immune system of the bacteria and archaea. But 100% bacteria do not have the CRISPR defense system. 60% or like that. So the probiotic bacteria which you are choosing as a candidate for commercialization, you may check whether it has, a robust CRISPR-Cas system in their genome or not. If it has a robust CRISPR-Cas system in their genome, by sequencing we can easily know it. Then it would be a nice candidate for fighting against the virus, and it can sustain, and virus cannot destroy your probiotic candidate anymore, because bacteria are most of the bacteria are prone to the virus, and virus has no way to proliferate except it uses the transcription and translation machineries of the living organisms, because virus is non-living particle in fact, and it cannot replicate of its own way, either RNA virus or DNA virus. So it needs a host, and use the host central dogma for making the copy of their nucleic acid, and then replicate their particles and this is really a fascinating and amazing system. In case of coronavirus, also it is doing the same job. Coronavirus is single stranded RNA. So when you register for any probiotic candidate, you must check it. Otherwise, bacteria, uh, probiotic bacteria that you are going to commercialize would face serious problem uh, uh, of bacteriophages. Any question?
So in my last day, yesterday, you have mentioned that
Roll, I mean, your sound.
a single capsule can. Okay, sir. Hear me, sir.
Yes.
Sir, yesterday you have mentioned that a single pharmaceutical capsule can contain around 2 billion probiotics bacteria. So far I know that this bolus are or capsule are in dry form. So how this bacteria will sustain or survive here? Um,
Yeah. This
or
is a
the
good question. In
can
case
you
of this, this is high tech they use. What they do, they encase bacteria in the nano film, encapsulation by nanofilm. So every bacteria is encapsulated by the polymeric substances, so that no stress from the environment can affect their survival. So this is another formulation technology because shelf life is very important. In the capsule, there is a deadline. Until that deadline, all the bacteria remain in the capsule or the tablet. They remain live. So bacteria are not directly used. They are formulated by using different polymeric substances, nano encapsulation or like that, and then bacteria can survive. And most of them, lactobacillus or other bacillus, they have ability to survive in the harsh environment. In the our gastrointestine also highly acidic and even high temperature is generated. So they have the ability to survive in ambient condition, that means room temperature. If you can keep them free from the sunlight or humidity and other factors, so they remain as they are sleeping there. So when you take that capsule, immediately they get the heaven, and they come out from the capsule and start to get their niche for colonization and exert the function. Any question?
oh sir. So far I can understand from your answer is that its capsule is actually this bacteria encapsulated by nanotechnology, is it?
Yeah, nanotechnology or microtechnology they use for making the formulation. I visited an USDA laboratory in Peoria. I saw how bacterial formulation, dry powder they are making. And in case of dry powder, maybe not a single bacteria, but a few bacteria, they are encased by the thin film of very thin nanofilm of polymeric substances that protect them from the environmental stresses, so that they can stay longer time. Bacteria even minus 80 degrees Celsius temperature in 10 to 20% glycerol, we can preserve for several years. Bacteria has such a capacity. It is another business to find out the best condition or formulation for commercialization of the bacteria to maintain their shelf life. We shall discuss later on also this topic.
Thank you so much, sir.
Any more question?
Okay.
No, sir.
Okay, then thank you so much.
Assalamu